Hello dear students, welcome to our class again, and today we will be seeing the fourth method of assessing the nutritional status, which is the biochemical assessment and the biomarkers which are useful in nutrition and health. We have already seen the earlier methods in the A, B, C, D, which is anthropometry, the biochemical assessment we are going to see now, and the clinical assessment and dietary assessment. Now, the nutritional status of an individual reflects the extent to which their physiological needs of nutrients have been covered at a particular life stage. When the nutrients to support daily body needs and metabolic demands are consumed in a balanced manner without insufficiency or excess, the person presents an optimal nutritional status. Then this favors growth, development, appropriate cell tissue turnovers and global health. Now, dietary assessment and nutritional status are traditionally measured by means of dietary intake data such as 24-hour dietary recall, food records or food frequency and questionnaires. So, even though recent technological advances including image analysis software to collect dietary information or to process dietary data have improved dietary assessment, food intake based methods have some inherent limitations such as subjective nature of data collection tools. The people do not always remember everything they have consumed or not able to recall all foods eaten or their specific ingredients or components, and they may have difficulty estimating the portion sizes accurately. This combination of factors determines the measurement errors in dietary assessment. Moreover, individuals often underreport dietary intake. They talk less about their dietary intake, particularly when reporting intakes which have to be higher social desirability and for those who have the history of dieting and being overweight reflect greater eating restraint. Then limitation of food composition tables. Some nutrients such as vast majority of the trace elements are not sufficiently characterized in the food composition tables, and therefore, nutritional status cannot be correctly assessed based on the intake. This is also the case for certain fat soluble vitamins. Fats and oils, they constitute the main nutritional source of vitamin E. However, the content of this vitamin varies depending upon the type of oil, its processing method and the addition of antioxidants and its shelf life. All of these which cannot be characterized in a dietary assessment. On the other hand, the nutritional content of the food is neither considered nor uniform, and food composition databases may not reflect the characteristics of the product that those are currently commercialized. They generally lag behind current eating patterns. For example, the tendency of the whole grain products is poorly reflected. Then factors influencing the nutrient absorptions. Certain nutrients have feedback control mechanisms that increase or decrease the efficiency of absorption depending upon the nutritional status of an individual. For example, if an individual with a low nutritional calcium status, he will absorb calcium more efficiently. Certain food combinations can affect the absorption. For example, if you have more fiber content in the diet, it may decrease the availability of food carotenoids, and vitamin C content promotes the iron absorption when it is ingested at the same time. So, you eat food along with some vitamin C, the iron content that is present in the food will be better absorbed in the presence of vitamin C. And vitamin D was shown to be better available from milk than from any solid food. So, the extent of cooking of foods also influence the composition, including the nutrient content and absorption. Such is the case of vitamin B6 and vitamin C. Sometimes overcooking may lose all these vitamins. Then finally, the degree of processing may affect the absorption. Such micronutrients can be associated with proteins that facilitate their bioavailability, which has been shown, for example, for calcium and zinc, or which are better available in their native form, that is heme, and the degree of processing may affect the absorption. Some micronutrients can be associated with the proteins, and these factors are generally not considered because dietary questionnaires do not include enough detail of how the food is prepared or processed, and they do not capture the information about the foods that are eaten together. Biomarkers are again an important method so that we learn how to assess the nutrients in the body. So, nutritional biomarkers, they can be measurements based on biological tissues and fluids and on physiological or behavioral functions, where more recently on the metabolic and genetic data that in turn influences the health, well-being and risk of disease. A risk biomarker is defined by the Institute of Medicine way back in 2010 as a biomarker that indicates the component of an individual's level of risk for developing a disease or level of risk for developing complications of a disease. As an example, metabolomics is being used to investigate the potential risk biomarkers for pre-diabetes that are distinct from the known diabetes risk. Risk indicators like glycosylated hemoglobin levels, fasting glucose and insulin. So, several laboratory based biomarkers exist to assess the individual's level of risk of developing the disease and as substitutes for chronic disease outcomes. They are often included as an adjunct to the clinical assessment. So, in combination of clinical assessment, if you assess the biomarkers or the physiological fluid assessment, then you can come to a better conclusion. Examples include serum ferritin for the risk of iron deficiency anemia and glycosylated hemoglobin for the risk of diabetes and alterations in bone mineral density for changes in fracture risk. Examples of surrogate biomarkers intend to substitute for chronic disease outcome, which include the LDL cholesterol instead of the true clinical outcome of CVD or cardiovascular disease and blood pressure for cardiovascular disease, which is a very clear example. The limitation of dietary assessment to estimate the nutritional status determines the need for analytical determinants that can objectively and accurately quantify nutritional status. Biomarkers can provide a more proximal measure for the nutritional status than the dietary intake. Generally speaking, the nutritional biomarker is a characteristic that can be objectively measured in different biological samples and can be used as an indicator of nutritional status with respect to the intake or metabolism of the dietary constituents. The biochemical analysis of a reference metabolite that indicates the bioavailability of a nutrient is an objective result to assess nutritional status, which entails lower methodological error and detects deficiency states more precisely than dietary assessment. Such biomarkers are generally based on pronounced changes observed in one parameter. They are clinically useful, in particular to detect the deficiencies in support of medical treatment. Analysis of folate, iron and vitamin B12, but also copper and zinc is useful to identify potential nutritional causes of anemia. The development of biomarkers faithfully representing the nutritional status for those micronutrients is clearly justified by their usefulness in medicine. Clinical biomarkers are focused on diagnosis of a disease state. In most cases, these are independent of nutrition, but comprise the best parameter that reflects a certain disease. In case of metabolic diseases, overlap may occur, but for example, this is the case for circulating lipid profiles, urea levels in the blood or urine and so forth. In most instances, clinical diagnosis of disease differs from nutritional biology that focus on health, that is, whether the nutritional status is such that it supports health or not. And the latter can also be entail mild subclinical deficiencies as well as moderate exercise. Especially in those cases, the combination of methodologies like dietary assessment by food questionnaires along with biochemical measures can provide a useful tool for estimating the exposure to a particular nutrient of interest and assessing the health risks. This combination may eliminate some of the errors associated with each type methods to assess the nutritional status. Examples of suggested nutritional biomarkers related with exposure and or effects of micronutrients, food or dietary patterns in samples obtained with non-invasive, minimally invasive techniques. Some representative sample are provided for each candidate. Biomarkers are shown in the table which show us the proposed biomarker, the sample type and the intended use for which the biomarker can be used. For example, alkyl isopronide, which is in plasma, and this can be tested when whole grain food consumptions, and some of it are like the carotenoids which can be tested in plasma, and this can be observed when fruits and vegetables intake is more. Similarly, carotenoids and vitamin C can be observed in plasma and serum when there is increased fruit and vegetable consumption, and combined marker can suggest both carotenoids and vitamin C. Then the creatinine level in serum can be seen by intake of meat and fish. In urine, the creatinine level shows again the intake of meat and fish. If we see the digoxin level in urine or plasma, the intake of soy based products can be known, and similarly the homocysteine plasma, you can see the carbon metabolism or the folate status. So, like this the various tests goes for, and the intake of different foods can be identified based on the test either in the urine or the plasma or serum. Now, nitrogen in 24-hour urine is an already substantially validated biomarker for protein intake. This type of review follows specific recent guidelines for the review, identification and validation of candidate biomarkers of food intake. So, to understand the complex relationship between the nutrients and the health, different types of biomarkers are being used in nutritional studies, which are markers of exposure or effect of the health, and disease state. Biomarkers of exposure. These include the different types of biomarkers used to evaluate dietary intake of nutrients, non-nutritive food components or dietary patterns. An example is the presence of nitrogen in urine. This serves as a biomarker of the protein intake. So, these types of biomarkers are of great interest as their use can help to improve the categorization of subjects according to the exposure of a particular nutrient. They also serve as an objective indicator of compliance with a particular dietary regimen in intervention studies, investigating the health effects of dietary modifications. So, these biomarkers may not only reflect one nutrient, but may also be associated with a dietary pattern or food group. For example, the plasma concentration of alkyl resorcinol is considered as a biomarker for the intake of whole grains, and a combination of sucrose or fructose with erythronic acid is a urinary biomarker for sugar intake. Urine or plasma genistein and diadzin are also biomarkers for soy and soy based product intake. While robust information for markers and other legumes is still lacking, in this sense a combination of the markers may better reflect a food category, and for example, vitamin C and carotenoids together may be more accurate if the fruit and vegetable biomarkers alone. And biomarkers of effects, if you see, these are the biomarkers that are related to target functions or biological response. Thus, not only do they reflect intake, but also the nutrient metabolism and possibly the effects on physiological and disease processes. It is important to note that a biomarker may not reflect the effect of a single nutrient, but the interactions of various nutrients. For example, some of the biomarkers of metabolism of one carbon compounds such as homocysteine, they reflect not only nutritional intake of but various metabolic processes related to pathological or physiological conditions. Biomarkers of health or disease and physiological status. So, these are the biomarkers which indicate an endpoint, relate to a state of health or disease risk. These markers reflect the different intermediate disease phenotypes or the severity of disease, and they are widely used in the clinical practice. For example, plasma levels of fasting glucose are associated with insulin sensitivity, and diabetes or plasma cholesterol and triglycerides are linked to cardiovascular disease. So, nutritional biomarker research is not focused on identification and characterization of diseases or treatment prognostics, which are the areas of intense development. Current challenges in the development of health biomarkers. The development of health or disease biomarkers was driven by medical needs and has largely been directed towards identifying and quantifying the disease states or progression rather than assessing the health status and quantifying the health status of an individual. However, main objective of the diet and nutrition is to promote and maintain optimal health. Therefore, it is highly relevant to have biomarkers of very early stages of alterations that may ultimately progress to the disease, even before what may be considered the onset of disease. Such biomarkers can be considered health and or prevention of markers rather than disease markers. Pre-disease psychological and physiological alterations are likely to be associated with pre-disease alterations in homeostatic balance and may be identified when a homeostatic response to a particular environment or nutritional aggression is examined. That is why the recommendation of having biochemical assessment every 6 months or 1 year is essential, so that we can identify the pre-disease alterations in the homeostatic balance. These biomarkers, they represent a new approach to biomarkers that reflect maintenance of physiological integrity and function. In this context, the health promoting food components support or even compromise and optimize a health physiology, preventing or delaying the initiation of disease state or a loss of physiological function, including cognitive function. Because of the multifaceted nature of homeostasis, nutrigenomic technologies which analyze functional genomic responses on a genome-wide scale applied to the field of nutrition have been particularly valuable and will continue to be so for the identification, characterization and validation of health biomarkers. Human health is based on a complex network of interactions between the pathways, processes and molecules, implying interactive mechanisms across different cells, tissues and organs. Various biochemical and physiological mechanisms are responsible for maintaining health in an environment that is constantly changing, and as a result, for example, diet, infections, temperature, exercise and various other stressors. In good health, the mechanisms that maintain homeostasis are able to effectively buffer the different challenges that the individuals are subjected to. The adaptation response defines the so-called phenotypic flexibility. The way to disease starts when and where these adaptive processes regulate the networks fail. Lifestyle and other conditions, both environmental and internal, can reduce the robustness and elasticity of these mechanisms. Then, the homeostatic machinery becomes less effective and they start producing negative side effects which can occur from the molecular level to the whole body level. An example is excessive accumulation of lipid in the liver, which can be a consequence of the diminished capacity of the adipose tissue for lipid storage. So, this results in an organism that has lost its ability to react adequately to the external challenges, which further aggravates the situation. For example, by the development of insulin resistance. This loss of adequate physiological performance, resulting in the inability to maintain healthy responses, can be the basis of the development for new biomarkers reflecting dynamic responses to assess the health status and the capacity for its metabolic flexibility. Biomarkers of health can guide policies related to food, nutrition and health. In fact, they would represent the basis of substantiate the health claims of food. At present, the lack of robust nutritional biomarkers may biological functions is a bottleneck that shows down innovation in the food industry. This is recognized and taken up by the scientific community. One example is the bioclaims, a collaborative research project carried out by the European level, which has established the principles to identify, establish and validate robust biomarkers to quantify the health status. So, examining the influence of bioactive compounds of the diet on these biomarkers provides a basis for evidence-based development of foods with reliable health properties and can contribute to healthier diet and health in the long term. And other initiatives like Preventomics. This is a project funded again by the European Union Program Horizon 2020 under the call of ICT. Personalized nutrition aims to use health biomarkers in the dietary advice applications for the consumers. So, Bond classified the nutritional biomarkers into three groups. So, biomarkers of exposure, food or nutrient intakes, dietary patterns, supplement usage, which are assessed by traditional dietary assessment methods, and dietary biomarkers, indirect measures of nutrient exposures. Then the second type of biomarkers are of status body fluids. The serum, erythrocytes, leukocytes, urine, breast milk and tissues like hair and nails can be examined. Then biomarkers of function, which measure the extent to which the functional consequences of a nutrient deficiency. It can be functional biochemical like the enzyme stimulation assays, abnormal metabolites, DNA damage, and these biomarkers serve as early biomarkers for subclinical deficiencies. Then you have the functional physiological or behavioral biomarkers, which are more directly related to health status or disease such as vision, growth, immune function, taste acuity, cognition, depression. These biomarkers, they impact on clinical and health outcomes. Now, functional physiological and behavioral biomarkers are more directly related to health status and disease than the functional biochemical markers. So, disturbances in these functional physiological biomarkers, they are generally associated with more prolonged and severe nutrient deficiency states and are often affected by social and environmental factors. So, their sensitivity and specificity are low. In general, the functional physiological tests, with the exception of physical growth, are not suitable for large scale nutritional surveys. They are often too invasive, they may require elaborate equipment, and the results tend to be difficult to interpret because of the lack of cutoff points. The growing prevalence of chronic diseases has led to investigations to identify biomarkers that can be used as substitutes for chronic disease outcomes. The chronic disease events are characterized by long developmental times and are multifactorial in nature, with challenges in differentiating between causal and associative relations. To qualify as a biomarker that is intended to substitute for a clinical endpoint, the biomarker must be on the major causal pathway between an intervention, for example, diet or dietary component, and the outcome of interest like the chronic disease. Such biomarkers are termed as surrogate biomarkers. Only a few such biomarkers have been identified for chronic disease, and low density lipoprotein cholesterol, the LDL cholesterol concentration within a saturated fat and cardiovascular disease pathway. Increasingly, it is recognized that a single biomarker may not reflect exclusively the nutritional status of a single nutrient, but instead be a reflective of several nutrients. their interactions and metabolism. This has led to the development of all-in-one instrument platforms that conduct multiple micronutrient tests in a single sample aliquot. So, a 7-plex microarray immunoassay has been developed for ferritin, soluble transferrin receptor, retinol binding protein, thyroglobulin, malarial antigenemia and inflammation status biomarkers, which has subsequently been applied to dry blood spot matrices. So, comparison with reference type assays indicate that with some improvements in accuracy and precision, this multiplex instrument platforms could be useful tools for assessing multiple micronutrient biomarkers in national micronutrient surveys in low resource settings. Readers are advised to consult the Micronutrient Survey Manual and Toolkit developed by US Centers for Disease Control and Prevention for details of planning, implementation and analysis, reporting, dissemination, use of data generated from National Cross-Sectional Micronutrient Survey. Now, coming to the biochemical assessment, this discipline involves the analysis of cells, blood and other body fluids for their chemical, biochemical and hormonal compounds. So, biochemical tests are most often applied to samples of serum, plasma and urine where the levels of specific chemicals are measured and the results are compared with those representative of a healthy individual. An increase or decrease in any particular component can help to identify a disease process. Like the measurement of blood glucose, that is sugar, lipids which are fats, are among the commonly performed tests in biochemistry laboratory. Panel of tests can be used to assess the function of major body organs like the liver, kidneys and heart. So, we have special assays that are used to measure the levels of various hormones in the blood. Biochemical and laboratory assessment methods are more objective, precise and sensitive methods for evaluation of nutritional status by any other method. It may aid in diagnosis of primary malnutrition resulting from inadequate food intake, and it is valuable in guiding to take necessary action in the secondary malnutrition which are resulting from conditions of increased need for or losses of substrate. However, the interpretation of the laboratory data is often difficult and does not necessarily always correlate with either clinical or dietary findings. An ideal biochemical test suitable for field survey should be sensitive, that is, easily identify most positive, and it should be specific, easily identify normal subjects, and it should be easy to carry out and preferably non-invasive and inexpensive. So, during the development of any deficiency disease, biochemical changes precede clinical manifestation. Therefore, biochemical tests are conducted to identify subclinical malnutrition or onset of clinical manifestation of a disease. Although biochemical estimations of nutritional significance can be carried out on variety of body tissues including liver, muscle and bone, in practice or field survey, the tests are confined to two fairly easily obtainable body fluids, that is blood and urine. So, laboratory tests for nutritional status assessment include measurement of a nutrient, its metabolite or some other product in blood and urine, measurement of the activity of vitamin dependent enzyme in erythrocytes and its in vitro activation with corresponding coenzyme. Then measurement of accumulated metabolite whose disposal depends on the vitamin or mineral dependent enzyme with or without preloading with a precursor. However, in a routine nutritional survey, all the biochemical parameters are not assessed. In general, biochemical and laboratory methods used to determine nutritional status are the serum protein, particularly serum albumin level, and the blood forming nutrients like the iron, folacin, vitamin B6 and vitamin B12, then the fat soluble vitamins, the vitamin A, vitamin D, vitamin E and vitamin K, the water soluble vitamins like thiamine, riboflavin, niacin and vitamin C, and minerals like iron, iodine and trace elements, and the levels of blood lipids like the cholesterol and triglycerides, glucose and various enzymes which are implicated in the heart disease, diabetes and chronic disease. But standardization of biochemical methods specific to a deficiency disease and standard reference values for cutoff points of the nutritional deficiencies have to be precisely defined. So, biochemical tests are conducted to detect marginal deficiency in individuals, particularly when dietary histories are questionable or unavailable. Their use is especially important before overt clinical signs of disease appear. This permitting the initiation of appropriate remedial measures and to supplement or enhance other studies such as dietary or community assessment among the specific population groups in order to pinpoint the nutritional problems that these modalities may have suggested or failed to reveal. Biochemical and laboratory assessment methods are more objective and precise, and they are sensitive methods for evaluation of nutritional status by any other method. It will help in diagnosis of primary malnutrition because of inadequate food intake. It is valuable in guiding to take necessary action for secondary malnutrition, where there is increased need for or losses of substrate. Ideal biochemical tests suitable for field surveys should be sensitive, they should be easily identified, they should be positive and they should be specific so that you can identify normal subjects, easy to carry out and preferably non-invasive and inexpensive. So, laboratory tests for nutritional status, they include the measurement of nutrient, its metabolite and the blood and urine are tested. They are vitamin dependent enzymes in erythrocytes and the activation of the correspondence coenzyme. As I told you, are present in the blood and the urine, and empowering citizens to monitor and follow a healthy diet. The future of the nutrition is moving towards the possibility of carrying out real personalized nutrition. The emerging concept is precision nutrition, which may be achieved as a result of rigorous nitrogenomic analysis that considers the genetic makeup of an individual and its epigenetic modulation and molecular phenotype. The health of an individual depends upon the information that is contained in its genome and how it is interpreted throughout its life, like epigenome or methylome, transcriptome, proteome or metabolome. This dynamic evaluation of physiology and the health status via integrated analysis of all these factors is what is called as integrated personal omic profile. So, although we are still far from being able to define this, the first description that exists of the IPOP for a single individual has shown enormous potential of omics integration in medical research, in monitoring the health status and personalized medicine. So, this is a preventive and diagnostic tool because it can follow and to a certain extent predict the evolution of health status and evaluate metabolic robustness. Furthermore, it also be helpful in improvement of the assessment of disease risk and provide high diagnostic accuracy, monitoring the disease, targeted therapies and understanding the associated biological processes. So, clearly the availability of such information requires powerful tools for integration and interpretation. Hence, it is necessary to do all the tests and come to a conclusion. So, the use of specifically designed platforms, for example, user friendly mobile applications capable of integrating all this available information, translating it into specific outcomes and expected to help empowering citizens to have healthier, optimal behavior and lifestyle adaptations. And there are certain biochemical test limitations. The tests are time consuming and expensive. They cannot be applied for large group of population, and to perform the test, experienced and well trained expertise are needed. Standard reference values and standardized specific tests for specific nutrients are needed. Most biochemical tests reveal current nutritional status only and are useful to quantify mild deficiency. It is an invasive method, so sample collection is sometimes hazardous. So, this is about the biochemical assessment and the biomarkers that are useful in the detection of malnutrition or deficiency diseases to the micronutrient level, and the frequent testing of the biochemical proportions of the blood or urine are very important to detect any deficiency at the earliest stage in the body. So, this biochemical test also is an important assessment in the nutritional status of the community. Thank you.